About things with the nucleus?
All right, so we are live on YouTube now, and we are going to start Jonathan's seminar. So Jonathan Klassen is a assistant professor at UConn right now. He had done his PhD at the University of Alberta and his postdoc at the University of Wisconsin, and has been at UConn for a little over 2 years at this point. And he'll be telling us today about leaf or ants growing fungus, right? And how that affects speciation, and I will let him explain better of what that means. So Jonathan, if you'd like to share your screen and take it away.
All right, let's see if we can make this thing happen.
And just a reminder, if you're watching, please feel free to tweet questions to MicroSeminar or use the hashtag #USeminar. If you're in the Hangout, we'll turn your camera and mic off and bring you back in a second, or at the end of the talk.
All right, how's that doing? Am I at the start?
Yep, go for it, and you can share your screen too.
Oh, okay. All right, let us share it. How's that? Have you got it?
Yep, got it.
Okay, fantastic. Okay, well, thanks, Jen and everybody, for having me. It's been really fun to follow these along, so I'm really glad to be part of it. So I'm going to talk about the work that my lab's doing with fungus-growing ants, and so there's a obligatory Alex Wild picture. If you do ant things, you got to have an Alex Wild picture. So I'll get that off the top. And I want to talk about species interactions because that's really what my lab is focused on, and how species interactions are important for structuring the microbiome and what the functional consequences of that. So I want to start logically with bison and wolves. And I like to start talks this way because it really highlights the difference between what big ecology does and what microbial ecology does. So I like this picture because it's pretty obvious what's happening, right? We have a bison, we've got all these wolves, and we can tell a couple of things. We can tell that there's an interaction happening here. These wolves are trying to eat this poor bison, and that's probably ecologically relevant, right? Because the bison dies. We can tell what's going on there, right? But we can also infer that these wolves, they're working together, part of the same pack, and so they're part of the same population, right? So this is why big ecology is great, is that you can see things, and that's awesome. So you take the microbial example, right? And so here's a picture of a field, and so the question is, how many populations are in this field? Right? How do you even think about answering that question? And one thing we know is if you zoom down to the super micro level, right, so this is a soil particle, and we know that sort of different sides of this particle can even be completely isolated from each other, right? And so those might host two different populations of microbes that never interact with each other in the wild, but how would we know, right? So of course what we typically do is we take all that soil, we grind it up in a tube, and we either do the 16S type thing, or we'll put it out on a petri dish or something like that, right? And so this one's the petri plate, and you can see where those arrows are pointing. There's an interaction happening there between two microbes. But because we've divorced what's on the plate from what's actually going on in the natural environment, we have no way of telling if that interaction is actually meaningful ecologically. So this, to me, is one of the main problems of microbiology right now, and sort of how I got into this whole story. So symbiosis is how me and my lab, we're working towards answering this problem. And I like symbioses because I think that they're really ideal models for studying species interactions in the microbiome. And the reason is that it allows us to take a bit more of a macro view of what's going on. We can make inferences about microbial fitness, ecology, population structure, because we can relate that to the fitness, ecology, and population structure of the host, right? So by working at using the host as a unit, we can talk about what's going on with the microbes. And there's lots of really nice systems that have relatively low diversity, which lets us really get into not get bogged down in the ridiculous complexity of a lot of microbiomes. And a lot of these systems are pretty amenable to experimentation. So I'll show you some of our work along those lines. So symbiosis is the one hand. Now the other part of my lab's research is to do with secondary metabolites. So these are chemical compounds that aren't part of what we consider core metabolism, so producing energy, producing biomass. But they do things that are more peripheral and are really often involved in interactions between organisms, right? So studying the evolution and diversity of microbial secondary metabolites, I think, really underpins the study of microbial interactions, because these are the traits that are driving interactions. So if we can understand what's going on with the secondary metabolites, we can start to understand what's going on in the communities. So this is a computational analysis that I did several years ago now, I guess during my postdoc, right? And there's two things I want to highlight out of this picture. So what you're looking at is a phylogenetic tree of all the bacteria that were in NCBI 3 years ago or something like that. Right, so a big multi-locus tree, and what I did was I took some software called Anti-Smash, and I annotated all the secondary metabolite biosynthetic gene clusters that were in these different genomes, right? So secondary metabolites, they often have their genes clustered together in sort of multiple operons. I mean, you can infer what flavor of metabolite a cluster might make from its sequence, and so that's what the colors represent. And so there's two things I want to highlight. One is you can go all the way around this circle, and pretty much everybody is making secondary metabolites. It's on the order of about 85% of the genomes encode for secondary metabolite biosynthesis. Right? So all microbes are doing this. Right? And the second thing to highlight is that this is a really colorful figure. Right? There's all kinds of different chemical diversity encoded by these different microbes, and really this particular type of analysis doesn't do that justice. The chemical diversity is massively huge. So these microbes are all interacting with each other using these types of metabolites, and there's this massive chemical diversity of what these traits look like. So that's something that really fascinates us. Okay, so the system that we work on is the fungus-growing or atine ants, and these have been really well studied for a long time, so well over 100 years now that these have been looked at. And they're most impressive in Central South America, the neotropics, because that's where the true leaf-cutting ants live. So these are the ones that have fascinated E.O. Wilson among many, many other people, and have really been sort of poster children for symbiosis for a long time, and also things like social biology and things like that, thinking about how the colonies are organized. Now what this tree shows is there's actually a pretty large diversity of fungus-growing ants, of which the leafcutters are sort of the terminal end, right? So fungus-growing ants, this habit of keeping a fungus, evolved once about 50 million years ago. Right, and then radiated since then, and it's only in the last 10 million years or so that the leafcutters, the really well-developed millions of ants per colony type of agriculture evolved. So there's this big diversity of more ancestral lineages, and we really don't know very much about them. But they're much simpler. They tend to use different nutrient sources, less contribution from fresh leaves in particular. And they're much smaller and simpler. So the one that we study is highlighted, Trichomyrmex septentrionalis, and that's what my lab works on. So it's just immediately basal to the true leaf cutters, and so it shares a lot of those characteristics. So Trichomyrmex septentrionalis is relatively small, so about 1,000 workers. So this compares to the leaf cutters in the hundreds of thousands to millions of workers. They do collect fresh leaves. I'm not sure they go up the trees, but certainly in springtime in particular, before the tannins have come out in the leaves and things like that, they'll take fresh leaf material. Mostly the things like oak catkins, so this is the seed pod of the oak, and caterpillar frass, or the poop of the caterpillar. So they'll use those types of biomass instead of fresh leaves predominantly. They're nice being so small. We can keep them in the lab fairly nicely and do lots of interesting things with them. So Trichomyrmex septentrionalis is the northernmost fungus-growing ant in distribution. So it ranges in the U.S. Florida would be the southeastern extent of the range, goes all the way over to Texas, up to Illinois, and then up to Long Island in New York State, so up by where I am. We've really focused on the eastern extreme of this distribution. So the map there shows all the places we've been. So we've done a lot of work in New Jersey, some on Long Island, North Carolina, Georgia, Florida, getting a nice transect across this gradient. And And one of the nice things about this ant is that it has a really well-defined population structure. So John Seal at UT Tyler has done a lot of work on this, and what his results show is that what seems to have happened is there was the Pleistocene glaciations, and it forced all the ants down into the southernmost parts of their range, and then they've radiated upwards from that. So it's a really a strong gradient of genetic diversity that's not super well mixed, and so we think that that's an interesting place to look at the microbial ecology and population structure as well, that it might follow similar gradients. That's a hypothesis, not stated fact at this point, but we think that there's a lot of truth to that. So so these pine flat ecosystems are actually pretty cool. They're interesting because they're fire controlled naturally. So ancestrally they were maintained by natural burns that would come through every few years. It would burn off a lot of the underbrush. And so that's a really helpful thing that we've been able to take advantage of, just to be able to find the ants. The fires are relatively cool. They don't harm the ants, but they take out some of the plant diversity. So that's good for us. Okay, so when we walk out into one of these forests, this is what we look for. So the shovel that you're seeing there, shovel head's about 6 inches across. They're little teeny tiny shovels, right? And you can see all the pine needles around it. But there's these pie-shaped colony mounds, and the hole will be right in the middle of that. Right, and this is one of those Trichomyrmex septentrionalis mounds. So when we find one of these trachea colonies, we'll stick a pine needle down there and see what comes out, and we can morphologically identify the ant as well. And then this will be what we collect. So the way that we do that, so here's a couple of people from my lab. So Kevin Lee's in the background. He's my PhD student. Lee Deininger's in the foreground. He's an undergrad transferring to a master's, and you can see the burned area, right, nice and charcoal on the bottom there. So a lot of the underbrush has gone away. You can compare it to the forest in the back. Much more difficult to find colonies in that. And so we're able to dig these things out. And so what you do is you start about a foot back from the colony mound, dig 2 or 3 feet down into the soil, and then gradually work your way toward the colony opening. And eventually, you'll hit a hole in the ground. And you do it really carefully so you don't knock any dirt into the hole in the ground. But what's in that hole is actually the ants' fungus garden. So the ants, they'll collect the leaves and caterpillar frass and whatnot, bring it down whole, feed it to a fungus, and then the fungus is what they eat. So in that hole, you'll see something like this. And so this is a fungus garden inside of a hole that we've carefully excavated out. You can see there's some roots around it. These fungus gardens are typically suspended from the little rootlets, the very small roots, and so are actually hanging down. And you can see they've got a very well-defined structure. That's actually the ants are constructing that. They'll incorporate the plant biomass on the outside, wrap the hyphal filaments around it, and the colony will digest it such that the older things go toward the middle and the bottom. But at the same time, the ants are moving around this quite a bit, so there's a lot of homogenization of the microbial populations that still happens. So we get in there with our sophisticated sampling equipment, namely a spoon. Using good sterile technique, flame your spoon in the field, and we can take out this little ball of fungus and transfer it without getting it very dirty. So, yes, we are in the forest with pill bottles and flaming spoons and things like that. We've only been aroused the authorities once. My poor undergrad was working at a picnic table and doing nitrogen fixation experiments as well. So she had needles and all sorts of things, and one of the New Jersey state troopers is behind her. What are you doing, ma'am? So that's the importance of getting your permits out. I, of course, had my head in a hole somewhere. I had no idea this was happening, but poor undergrad. So always have your paperwork in order is the moral of that story. But we meet a lot of great people in the forest. They're always very helpful once they find out what we're up to. So the other thing we can do is we can collect the individual ants themselves. So this is an instrument called a pooter, which is basically a tube attached, 2 tubes attached to a collection vessel, and you suck in one end, and the ants go in the other, and they get stored in that 50 mil Falcon tube in there. So we're able to collect all the ants along with the fungus garden and bring these things back to the lab, and that works quite nicely. So in the lab, then, we've got a room full of ant colonies. So each of these green trays here will house a different ant colony, and so from all the way up down this East Coast transect that we've done. And you zoom up to one of these, and this is what it looks like. So what this is there's a plastic box there, and there's plaster of Paris down along the bottom, and the fungus garden is situated on top of that plaster of Paris. There's a tube that goes into the plaster of Paris, and a couple times a week we fill that up with water. The water seeps into the plaster of Paris and slow releases, keeps everything nice and moist. And then you can see there's another tube on the right that connects to a separate box. The food will be in that separate box, so the ants will go collect the food, bring it back, incorporate it into this fungus garden, and then they'll take out the trash as well. So they'll move the trash into the second collection box. So it's a really nice system. I mean, this is basically what you would find in the wild, a ball of fungus about this size. These particular boxes are probably 8 inches by 8 inches square, right? So not very big. And we can get most of the ants in the colony in this as well. So we can keep the system going in the lab quite nicely. So that's a huge asset. Okay, so what I'm going to do is I'm going to build up this figure of how this symbiosis works. So I've introduced you to 2 partners now. So the first partner being the ants, and the second one being the cultivar fungus. And so this particular fungus, there's only a very specific lineage. It belongs to the genus Leucoagaricus, although the taxonomy is messy. But it's a specific fungus, and the fungus provides the ant food, the ant provides the fungus food, and everything, it's a nutritional mutualism there. So a little bit more about these cultivar fungi. So they're obligate symbionts of the fungus-growing ants in Trichomyrmex. You don't find them outside of the symbiosis. That's not entirely true of all fungus-growing ants, but is true in our case. One of the things that is specialized about these fungi is they have these hyphal swellings at the sort of terminal ends of their hyphae. These are called gongolidia, and you can see in the figure on the left, or sorry, on the right, these swellings on the end. So this is magnified about 400 times and stained with Congo red. And at the tips of the hyphae, these big swellings, and those are chock full of nutrients, and that's what the ants collect, and that's the ants' food source. And there's a single strain of cultivar fungi maintained per ant colony, as far as we know. So why single strains? And that brings me to another really interesting part of the biology of these ants, and that is the reproductive cycle. So social insects, these ants have one queen who does all of the reproduction, right? So she lays all of the eggs. They develop mostly into sterile female workers, and then when the colony gets big enough, they'll begin to develop into males and females. These are winged sexual states. Right? And they will leave the nest in a mass swarm and mate. Now, before the queens leave, they take a little bit of that fungus garden and put it in a pocket, essentially right by their mouth. Right? They fly off. They mate with the boys. The boys all die because that's what they do. The queen lands, loses her wings, and regurgitates that bit of her fungus from her natal fungus garden, and that's what she starts her next garden from. So you can think of this like propagating an E. coli culture, right? You take a little bit out, you put it in a whole bunch of new broth, you grow up a huge vat of E. coli, right? But you're only taking a very small percentage of what you started from. So there's a serial dilution that happens here each time, and a really strong bottleneck that happens, and that we think that's part of what maintains really low diversity in the system. Okay, in Trichomyrmex septentrionalis, we find there's probably about 4 different cultivar, I'm calling them ecotypes. They belong to the same phylogenetic lineage, but are sort of distinct at the strain level. They're morphologically a little distinct, especially how they form their gongolidia and branching patterns of their hyphae, and they appear to have different secretions. So this is sort of a first indication that secondary metabolites might be involved from the cultivar fungus. So in the picture, I've shown 3 different types of this, all on the same media, and you can see they have all discolored the media to various extents, and they look morphologically a little bit different. So there's some diversity at the cultivar level, but 1 strain per colony, there's just sort of different possibilities that a colony might have. Okay, so one potential problem with this system is that if something's able to infect and kill this cultivar fungus garden fungus, that's bad news for the ants, right? And mycopathogens are widespread in nature, and indeed in this system, there's been a lot of work by Cameron Curry's lab, so my postdoc supervisor, who had worked on this one fungus in particular called Escovopsis, and it's a real specific pathogen of this garden fungus. So in the picture here, you have on the left-hand side is a healthy garden. It looks sort of grayish. On the outside, that's fresh biomass being incorporated in. On the right-hand side, there's this white fuzz, and that's the Escovopsis fungi who've just devastated this particular fungus garden. So getting sick is bad news for the symbiosis. Now, we've looked at this in our species, and interestingly, we don't find a whole lot of Escovopsis. It's quite rare. We've only found it in Florida, and we think that perhaps there's a climactic limit for how far north it can go. So there's a geographic structure to fungal pathogens in this system. What we do find instead is that there's a wide variety of what we call more general fungal pathogens. We don't have any evidence that these are specific, and they're usual suspects, right? So things like Trichoderma, Fusarium, Penicillium, Eucor, these are pretty common mycopathogens. And these are what we isolate a lot. And we think that this is the main disease burden in most of our trachea colonies. And you can see the percentages just in terms of frequency that we isolate them. So our undergrad army working hard on that. Okay, so if I add this to the interaction network that I'm building up, we have the ants, we have the cultivar fungus, and then we have these various fungal pathogens. And so I've drawn a solid green line between the cultivar fungus and the fungal pathogen. That indicates that the fungal pathogens grow on the cultivar, and then a solid red line going the other way. That when the fungal pathogen infects the cultivar, that's directly bad for the cultivar. And then I've drawn an open arrow, red arrow, pointing toward the ant from the fungal pathogens. And that's to indicate that when the cultivar fungus gets sick, that's indirectly bad for the ant because it loses its food source. Okay. So one of the really fascinating things about this system is that one of the things the ants have done to combat this pathogenesis problem is that they host another microorganism, an Actinobacteria, on their cuticle. And these are pretty much invariably belonging to just a few lineages of the genus Pseudonocardia, and they produce secondary metabolites that have been shown many times to protect the fungus garden against fungal pathogens. So on the right-hand side, I've got a picture of an ant taken by a really wonderful macrophotographer, Mark Smith, who we've collaborated with. He's got a great photography system where he's able to image stack, change the focal plane gradually and sort of zoom in and out, and so you're able to get a really wide focal plane with super high magnification. And so he's imaged our ants for us, which has been great. And you can see these white dots just below what would be the ant's neck, the propleural plates. Propleural plates, difficult to say. And if we zoom in on those, you can see that each one of those is essentially a colony of Actinobacteria. And so we can take these, sample this Actinobacteria very specifically by holding the ant upside down, scraping that particular plate, and more recently we've been dissecting those plates off as well, right? And you can see each of those dots represents a colony of Pseudonocardia. So each of those dots, we think, represent an individual crypt, and these crypts occur all over the ant cuticle. So this is work from Cameron's lab some years ago now. And the degree to which that white coating will cover the ant varies depending on ant age especially. But in adult ants, it's principally localized on the propleural plate, as you've seen. And in each of these crypts, you can see in the image, right, the image shows the crypts with the bacteria stained with crystal violet, and there's a gland that occurs at the bottom of these crypts, and the ants are presumably feeding the ants from that gland. We don't understand very much about how that works, but it appears that the ants are feeding the Pseudonocardia, and that's the basis for maintaining that defensive mutualism. Okay. Now, one of the interesting things is, just like the cultivar fungus, to the best of our knowledge, each ant colony only hosts a single lineage of Pseudonocardia. And we can think of this in two different ways. One is that just like the cultivar fungus, the pseudonocardia is transmitted between colonies primarily by the queen, who's got pseudonocardia that she takes with her when she founds a new colony. The second thing is that it's predominantly vertical transmission that occurs within a colony. And this is because when new ants emerge, they're helped along by the other workers in the colony that already have pseudonocardia on them, and that's a principal source of inoculation. So I've got a couple more images here. So this is a trachea larva from one of our colonies, and you can see that there's this membrane on the outside, and really what's in the inside there inside that membrane does not get colonized bacteria. So the larva will develop into a pupa still surrounded by this membrane, and when the pupa closes and becomes an adult ant, that's the first time that it'll be exposed to bacteria from the outside. And because it's being helped out of this casing by the other adult ants in the colony that have pseudonocardia, that's how they're principally inoculated. So really interesting mode of not quite vertical, but almost entirely vertical transmission. Okay, so we've taken all the colonies that we've sampled and we've asked, is it all the same lineage of bacterium in pseudonocardia bacterium in all of these colonies from this species? And we find that that's true, at least to the extent that 16S can tell us. So the tree on the right is just a basic 16S tree. I've labeled 6 different clades from previous work that I've listed here of what the diversity of pseudonocardia and fungus-growing ants looks like, and all of our Trachomyrmex pseudonocardia all fall into one clade. So this is consistent with some specialization occurring here. We've started to do genomes with these, and we spent actually a lot of time working on library prep for Illumina sequencing, which my Curie Lab colleagues can tell you, confirm has been a real problem. And I think we finally got that licked. Really, it seems that using library prep methods that include PCR is really bad news for these genomes. So this is preliminary data, but you can see that of the 3 we've got, they all cluster very closely together, forming so far a monophyletic group compared to the other pseudonocardia that have genomes. And they're quite highly related to each other, 96 to 98% average nucleotide identity. So this is comparing all the nucleotides in the genome to each other. And so this is we would call them the same species, although recognize that there's genetic diversity within that. Now, when we look at secondary metabolite diversity, and that's what's shown here, so each of the boxes in the little heat map is a different secondary metabolite gene cluster, and they're all different colors depending on the flavor of the cluster. And you can see there's quite a lot of secondary metabolite biosynthetic potential in these genomes. So each genome has got in the 20 to 30 type range of clusters in it. And some of these are conserved throughout the genomes that we've sequenced so far. Some of them are only conserved in the ones that are more closely related to each other. And there's a huge number of them that are unique. And so we're going to keep expanding on this, but I want to call back to some work I did in my postdoc with Cameron Curry, looking at pseudonocardia isolated from a different ant species. So this is from the fungus-growing ant Actinobacteria, which is another one of these lower attines. And we've done the same experiment, isolated pseudonocardia from all of these Actinobacteria, in this case from Panama and Costa Rica. And each column in this figure represents an orthologous gene cluster. So it's probably producing the same or similar compound. And you can see there's the same pattern, right? There's lots of things there's a few things that are very highly conserved. There's some things that are more patchily conserved. And what you're not seeing is that this heat map actually extends about 3 times as far to the right of all the singletons. And I think I've only included ones that appear 4 or more times. So there's a huge diversity of secondary metabolite biosynthetic genes that these things encode, but they're not conserved very well. And there must be some mechanisms of maintaining this diversity, and that's something that we don't have the answer to, but we're working on. Some of the ideas we're kicking around as to why this might be maintained is, well, maybe the pathogens have resistance, and so you have to get new compounds to overcome that resistance. It's possible that there's things like balancing selection operating, so 2 different pathogens, each of which requires a different secondary metabolite to target it, and so both of those metabolites might be maintained within a given population of the pseudonocardia. Could be that a lot of this diversity is neutral. It's just passing through, and eventually sometimes things stick and sometimes they don't. And it could be that they're targeting a much broader range of targets than as we used to think about it anyway, principally targeting Escovopsis. Maybe it's broader than that. And I'll note that we don't find Escovopsis throughout our sampled transect, but we find the Pseudonocardia throughout our sampled transect. So it's possible that the Pseudonocardia have been maintained just because they haven't been lost yet, but I don't think our data supports that. I think it supports more that those Pseudonocardia are targeting something else, perhaps in addition to the Escovopsis. Okay, so I add the Pseudonocardia to my little network here. So Pseudonocardia is probably fed by the ant through those glands that I showed you. So that's a solid green bar from the ant to the Pseudonocardia. And the Pseudonocardia negatively affects the fungal pathogens, right? So the solid red bar. That indirectly benefits both the ant and the cultivar, because if you kill the fungal pathogen, you protect the cultivar. If you protect the cultivar, the ant has food. So adding more links to our network here. All right, so the last bit that I'll add here is something we've spent a lot of time over the past couple of years, and we don't have the full story here, but I want to at least show you how this is shaping our thinking on how secondary metabolites are working in this microbiome. So it turns out there's a significant number of bacteria that live with this cultivar fungus. And there's been some work done on this by folks in Cameron's lab, but we really tried to build on this and expand this. So this is work by Frank Aylward, who was a PhD student in Cameron's lab. Now he's in Ed DeLong's lab. Frank did a bunch of metagenomes from the true leafcutters, so and Acromyrmex. And he found a really interesting signature of really limited diversity, really dominated by Enterobacteriaceae. Right? Some of these taxa appear to be fixing nitrogen. There's some evidence that this might be passed to the cultivar fungus. So, but the idea is that these are potentially nutritional mutualists of the cultivar fungus. Right? What's really fascinated me about this is that these are all taxa that we've been pretty good at culturing historically. And so we've done a lot of work along those lines to try to ask the same sort of questions we've been asking with the Pseudonocardia. From the genomes, so from those same metagenomes that Frank had done, and also from a number of genomes that exist for these bacteria, we can see that they encode for a wide variety of different secondary metabolites and a pretty substantial diversity of these metabolites as well. So this is another reason why we think, well, there might be some interactions mediated by secondary metabolites that involve these taxa, and so this is something we've pursued. So our isolations, we do find particular clades of certain bacteria that pop out again and again the more colonies that we sample. So I'm showing you here a particular clade of Pseudomonas just as an example. So these are candidates to be true symbionts, mutualists that are persistently associated with the system. And we were really fascinated to see, even just in our basic smush up some fungus guard and put it on a petri dish, that we can see bacteria interacting with each other antagonistically just from that simple experiment. So this is a fungus garden that we took out of a forest in New Jersey. We smushed it all up, we plated it out on a petri dish, and you can see there's these larger white colonies that are clearing out the smaller ones. So this is confirming, supporting this hypothesis that there's something going on involving secondary metabolites between different fungus garden bacteria now. We've been doing a lot of bioassays, so this is a really simple setup we've used, a streak plate assay. Basically, you take one focal strain and you streak it down the middle, and then you take different test strains and you streak them on the side. And you can see where all the eyes are on the right-hand side of the figure, that there's inhibition happening there. And what we see is a very high hit rate of these fungus garden bacteria that are inhibiting each other. And actually, one of the really fascinating things from a drug discovery perspective that I won't go into here, but it looks like there's a really substantial activity against the bacteria that are really closely related to known human pathogens. So the escape pathogens are a group of pathogens where resistance is to antibiotics that are clinically used is a real problem, and that's something we've really been targeting with these strains because we seem to have a very high hit rate against these potentially clinically relevant strains. So we're continuing to pursue that. Now, one of the interesting things we observed is when we do these same sort of bioassays, now using the pathogen fungi paired with the fungus garden bacteria, because we hypothesized maybe they have a defensive role as well, we saw really diverse effects, and we need to do more of these bioassays, but I think it's fascinating that we can get both increases in fungal pathogen growth and decreases in fungal pathogen growth depending on what strains we're looking at. So there's a lot going on here, and I think the interactions mediated by fungus garden bacteria have the potential to be fairly complex, even though their diversity is fairly low. So interesting stuff. Okay, so that's all at the bacterial level. We've also started looking at the metabolomic level, so really looking at the molecules, not the taxa that produce them per se. So we've really begun using this tool called metabolic networking, and we've done some of this in collaboration with Peter Dorstein's lab at UCSD, who's really been the pioneers in developing this technique. And so the idea is that you take some sample and you extract all the metabolites out of it. So we do a dichloromethane methanol type extraction, so we're getting things that sort of aren't super water soluble, but that look like secondary metabolites. You put these things in a tandem mass spec, and you get a whole suite of data that show both the masses that are present in that typically environmental sample, and also shows fragmentation, right? So each of these the parent masses are broken up into smaller chunks, right, depending on how they fly in the mass spec. And so you can take those data and you can organize them into networks. And so I've got a dummy network down here. Each of those dots is a node, right? And in our metabolic networks, each of those dots, those nodes, is going to be an individual parent mass, so a different compound in our network. And then we'll draw a line between them if those two parent masses, those two compounds, share a mass fragment. So maybe one has a sugar group that was lopped off and the sugar group's not on the other one. Well, they'll share the rest of the compound, and you can see that in the tandem mass spec data, and so you can link those together. So this allows you to characterize the families of chemical diversity that are present in a sample, really at a systems type level. And what we're really excited about is because we have all these pure cultures as well, we can take the systems level data, we can do the same thing on our pure cultures, and we can start mapping them on top of each other to really tell what's going on in the wild, and then when we take it into the lab, right, we match to those wild profiles to figure out who's doing what. And this is becoming a real focus. And so this is an example of what these networks look like. So a reminder, each of these dots is a particular chemical compound, a particular molecular mass, and they're joined together if they share substructures, essentially, right? So we see lots of metabolites. This is all generated from a single fungus garden, and I'm not showing you things that are singletons, that don't have any fragment that matches. And so this is done in collaboration with the Dorstein lab, and also locally with Marcy Balunis's lab here at Yukon Department of Pharmacy. Great natural product chemists who we collaborate a lot with. I'm going to focus in on just a few components here in this network. And so you read the dots this way. So we took one of those fungus gardens, extracted all the metabolites out of it, and all those metabolites are in the green squares. Okay. Now we also cultured out the cultivar fungus from that same fungus garden and grew it in the lab, and then did the same chemical extraction, right? And each of the metabolites present uniquely in that cultured fungus are in blue, and then in yellow are the compounds that were present both in the wild collected environmental fungus garden and the cultivar that we cultured in the lab, and you can see there's lots of places where they overlap. And that is the most fascinating thing to me, is that we can pin down these match compounds to who's making them, and I think that's a huge advantage of this model system in particular. So we see lots of things that in this case the pure cultivar fungus is making, right, and that those are present in the wild collected community. But there's also some other things that are in the wild but not in the lab, right, and those masses are perhaps coming from food. There's certainly some in there that are chlorophyll and things like that. But there's lots of things that we think might be coming from other microbes present in the fungus garden. And so we're tying all of these molecules to the taxa that make them. that's definitely a way forward. I've marked one dot here with an asterisk just as an example. And so this one compound has a match in the database because you can take these molecular masses and say, well, what else do we know that has it? And so the one that I've marked with a star there is this corticosteroid compound. We have no idea what it does. It's never been reported from these fungi before, but it looks like they're making a whole family of corticosteroid-like compounds. So so now we can take these compounds, look in the Sigma catalog, see what we can buy, and start testing them. Does it have an effect on the ant? Does it have an effect on these different microbes? And we can start pinning down really at the molecular level what's going on. So a great way to couple the survey with the detailed work. Okay, so this is the way we think of it, is that this microbiome has a number of different partners, a fairly limited number compared to, say, the human gut microbiome or something like that. And natural products are really mediating a lot of these interactions that are happening within this microbiome. So this is how the world works according to us, which of course means it's right? Okay, so one thing you'll notice I haven't shown you yet is the obligatory microbiome 16S stacked bar chart, right? And it turns out that that's a horrifically difficult experiment to do with this system. I had no idea. First PhD student came in and, okay, Kevin, we're gonna do just some 16S. We're gonna see who's there. It'll be easy. It'll be great. This will be done. Kevin went off. All right. Turns out that fungus garden DNA, especially from bacteria in Trichomyrmex, is horrific to extract. So the type of extract that Kevin makes is shown in the upper right there. He basically came to me for about 6 months saying I made more stout, and eventually sort of became more tea-like as the quality of his extractions improved, and he tweaked his methods. But there's no way we could PCR 16S out of any of these samples because there's so much PCR inhibition, and that's what the gel shows. He's got his positive control that works. Whole bunch of test samples, not a thing. But the other problem is that we found that the bacterial DNA, because there's so much fungal biomass from the cultivar, that it essentially swamps out our ability to PCR amplify the bacterial DNA because it's present in lower abundance, we think. And when we went back and dug through some previous literature and deep in the supplemental data, other people have noticed this, but no one really said anything or dealt with it. And we kind of got stuff eventually, so that's what we report. So Kevin, bless his heart, has done a lot of work really tweaking this, and he's come up with his magic DNA extraction protocol. And it's a bit of a beast, but it works really well. So I'll show it to you here. So first thing he does is he has to do a buffer wash to get rid of the most water-soluble inhibitors, and he spins it down real hard, so none of the bacteria come out, but he's able to take off a bit of those stout compounds, right? And then he went into the old literature and was able to find some really old DNA extraction methods that actually physically separate bacterial biomass from fungal biomass. And you can use differential centrifugation. You spin at different speeds, you can pellet out one versus the other. And you do this over and over and over again. And actually your recovery is pretty good and pretty unbiased. So that works pretty well. So now he's got his bacteria separate from his fungi. It's not just bacteria. He's got yeasts in there. We've saved different fractions because they might have viruses in them and things like that. So Maya someday will figure this out and will make you help us with that. After that, he does a standard bead-beating chemical lysis of the bacterial fraction, cleans the whole thing up with Ampere beads, and then loads his PCR up with BSA. And in the end, this seems to do the trick. So all his cleanup steps here have really reduced the PCR inhibitors. So on the left-hand side, I'm showing you a UV vis scan of one of his DNA extracts, and the top line, the solid one, the dash didn't show up. Well, the top line is one of these extracts, and you can see it's just chock full of inhibitors, especially in the 200 and below range, and also some stuff at higher wavelengths. Cleans it up, he gets rid of a lot of that junk. But you see his PCR on the right-hand side, the 174 lines, there's still no PCR product there where he's expecting it to be. Once he throws in BSA, he's finally able to get something, and we think the idea is the BSA is binding those residual PCR inhibitors and letting the polymerase do its job. So after 6 months, a year, Kevin, of really hard work from a really good student, Kevin's finally able to do this, what we thought was going to be this simple experiment, right? So I want to remind everybody what the expected results were going to be. We are expecting from the leaf-cutting ant metagenomes that are going to be dominated by Enterobacteriales type organisms, gamma proteobacteria. So what did we get? We got nothing of the sort. So this is the type of thing that you just, in this project particularly, after all that work, you just kind of sigh and sit back down for a second. What he found was his communities were almost entirely dominated by Clostridia when he extracted from freshly collected Trichomeromix fungus gardens. Right? This really strongly differs from previous data and is really counterintuitive because these fungus gardens are aerobic, right? These fungi are respiring aerobically, right? And that's how they're doing their business. I mean, you saw the lab colonies that we maintain in the lab, right? There's not a whole lot of opportunity for anaerobic microsites, at least relative to the aerobic parts, that's for sure. And we're able to culture tons of aerobes out of there, and previous data shows they're aerobes. So what's going on? Well, we have no idea. So one of the things Kevin did is he took one of the samples that had been sitting in the lab and being maintained actively in the lab for about 6 months. So he processes one of those samples, and lo and behold, there's our Enterobacteriales. The Clostridia go away. What we expected to come up comes up. And so we both give a sigh of relief, but then, well, what's going on, right? So we've got some theories, and we're at the point of testing these right now, but this is what we think. So one possibility is there's a zoo effect, right? By taking these microbes into the lab, that it's changed where they normally live, and so maybe that's changed all the conditions. Well, we think this is unlikely because natural fungus gardens are known to be aerobic, and previous data and all those sorts of things. Perhaps there was contamination during collection. Well, it's a possibility, right? We do our best to keep all the dirt out, but there's always the possibility of some getting knocked in. But I hope you could remember the picture I showed you. I mean, we're able to keep those fungus gardens pretty clean and pull the whole thing out pretty neatly. And the other thing is that soil microbes are not just Clostridia. They're typically much more diverse. So the profiles we're getting don't look like soil. And Kevin's in the process of showing that experimentally, definitively, but we don't think soil is the main problem. So this is our working hypothesis, and our hypothesis is that fungus garden microbial communities, at least in Trichomeromix, are really naturally structured by immigration. Okay, so what I mean by this is that there's a constant influx of these Clostridia. And they may not be functionally important, they may not be active, they certainly appear to be transient, but they're the primary members of these microbial communities in terms of abundance. Right? So where are they coming from? Maybe the insect frass. I mean, there could be some contribution from soil, so we're waiting for the ground to thaw and the snow to melt and the ants to become active again so we can get out there and muck around in the dirt and test this. That's what we think is going on with these microbiomes. So I'll add this to the figure, right? That we've got all of these microbes from the soil, from whatever else, and that's coming into the system. And so the question is, what's the effect of this? Right? What's going on there? How do we draw the arrows to connect this to the rest of the network? Well, it turns out if you go into the literature, which we did at this point, that there are a number of microbiome communities that are similarly really strongly defined by immigration. So the stomach is a great example, right? That 99% of the microbes that are going into the stomach are coming from the food. They're not coming from, they're not indigenous to the stomach. So if you did the analogous experiment with something from a human stomach, you would see the same sort of idea. So it raises questions then. Why don't the immigrants displace the natives? Right? It's possible that the natives are adapted to a niche that the immigrants aren't, so in particular the oxygen thing in our case. But that doesn't work in all cases, right? Should be more things coming in than just Clostridial spores, even if it's from insect frass. We know there's other things that live in there, right? So what's going on there? One of the things that we think is that there's a colonization resistance thing happening, and so it begs the question: do high rates of immigration impose a cost? Right? Is there what does fitness look like in these types of environments? And one of the hypotheses that we're working on is that the secondary metabolites that, in particular, the fungus garden bacteria might be making and that the cultivar might be making might serve to defend against these immigrants. Right? So there's a fitness and metabolic cost that these organisms are bearing just because of this high rate of immigration. And I think it's a really interesting idea. I think that when you look at ecological, like meta-community theory and things like that, that the actual cost of immigration we know remarkably little about. And so we're really looking forward to continuing to test this. And it really begs the question then, what's a microbiome anyway? Right? You look up the definition of a community, and it's a bunch of taxa that are in a place. Right? When we talk about microbiome, we often use it slightly differently. Right? You read a lot of the human microbiome type papers. First line is humans and their microbiome have co-evolved over millions of years. Well, if the stomach's 99% immigrants, that statement is probably wrong, at least for the stomach, right? So immigration is likely obscuring ecological patterns, and is going to we have to take that into account when we're thinking of evolutionary scenarios and mutualism as well. Okay, so I'm going to conclude there. I hope that I've made you think about our view of the world, microbiomes as networks of interactions between different taxa, and that there's a real need to account for this in models of mutualism. And one of the things that we've really been thinking about and are writing some papers around right now are that the biotic components of shaping a host niche are really important, right? So that microbes associated with the host they really cause a shift in the niche that host is able to occupy. And you can say the same thing reciprocally, that the host shapes the niche of the microbes. And the biotic components are all of these interactions working together. I hope I've convinced you that secondary metabolites are key traits that structure these communities, and that by doing so, they almost certainly help define how these communities function. So in our case, as defensive against pathogens is a particular function we've looked at. And then the last thing, I hope that I've made you think about how immigration might be, in some cases, a really dominant force that structures microbial communities. And when we do microbial ecology, we need to be really conscious of accounting for all of these different potential ecological dynamics that could structure what we're seeing in our data. Okay, with that, I'd like to thank my lab. So the folks there have done a truckload of work. So Dr. Sarah Kopac's my postdoc, she's done a lot of work on fungus garden bacteria. Sarah Goldstein's done a lot of work on the pseudocardia. Kevin Lee has done a lot of the molecular metagenomics and barcoding work. Emily's a new master's student. She's going to work on the ant host side. And then we have a huge army of undergrads who have done all kinds of different work, everything from the chemical extractions to isolating millions of bacteria, not millions, but we're at thousands, that's for sure, figuring out who they are. And then we have a huge raft of collaborators. I've listed the ones whose data I've shown. In particular, right now, Marcy Balunas is helping us with the chemistry locally, and then Peter as well, and Cameron and John Clardy's lab for things done more affiliated with postdoc, but continuing collaborations as well. And then definitely acknowledge funding that I've been fortunate to receive both from the JGI and from UConn. So with that, I'd be happy to take questions from people.
All right. Thanks for an awesome talk, Jonathan. I loved the what is a microbiome anyway portion of it. Talk more about that. So a few questions came in, and people are in the Hangout. If you want to jump in, but I can start asking the first few questions. So one of the first questions we had was actually a methodology question in terms of how did you make the really cool tree, I think that was of the metabolic categories?
The one that's got all of the metabolite, the gene clusters all around it?
Yeah, secondary metabolite one.
Yeah. So when I was in Cameron's lab, I had coded up a pipeline that we called ORFCore, and it's got some components in it that'll pull out 31 different marker genes, concatenate them, and build the concatenated phylogeny, and it's multi-threaded and all these sorts of things. So it works pretty well. We can handle a lot of genomes. And then the anti-smash software is what we use to do the annotations of the secondary metabolite gene clusters, and using the ITOL site, we can map those on as a heat map, essentially, onto the tips. So it can be done. It's all right.
So the other question was, So on your difference between the Clostridia and then the sort of native population that had more Enterobacteriaceae, and David, actually, if you want to jump in, it was your question. I know you've got kids there. That he was asking do you think that because of this harsh extraction, how you had to go through multiple steps to sort of purify things away, is there any chance you actually selected for a sporulating organism?
Yeah, we've thought a lot about that as well. One of the things we've done and we're continuing to work on this, right? But looking at how the microbes we're getting out with each increasing fractionation, how they change going that way. And one of the things we've been really worried about is the potential for there to be biofilms on the fungal hyphae, and so those being the most difficult to detect. But the same methods were used for both the one that generated the Clostridia and the ones that were primarily Enterobacteriales. The only difference there was if the material came fresh out of the ground or had been maintained in the lab for a while. So I think that even if we are biasing towards the spore formers, that the bias is the same for both of those samples.
So you use the same exact method with the lab?
I'm sorry?
You use the same exact extraction method with those labs?
Same exact method.
Yeah. Does that answer your question, Dave? Okay. Anyone else in the Hangout have a question? If not, you can tweet them to us if you're watching online.
I think Maya's got one.
I got one. I can't handle tweeting, but I can wave. So when you did the culturing of the fungi to get the metabolites, you had a lot of metabolites produced. Like, are those just produced kind of just constitutively, or were you actually challenging them with something that they would want to fight back against?
Yeah, great question. Those data that I showed there were just constitutively.
Wow.
I think it'd be more analogous. I showed a picture of the 3 different plates, and they all discolored the media to different extents. I think those are the metabolites that we're seeing, but definitely the challenge is the next set of experiments. So, yeah, that's what we're doing.
Also, Florida's not frozen if you just want to come down here and sample.
Oh, yeah, super interesting. So we came down in Florida a little bit over a year ago. In November, and we just caught the ants going into torpor. So what they do throughout the whole range is that they'll go into a torpor, a stasis, over winter, right? And so very pronounced in Jersey and places like that. But even in Florida, we found some colonies were really active in November, and some of them we'd shave down the tunnel, and they'd all be bunched up actually in the tunnel connecting the mound to the garden. They'd just all be in there, just hanging out, right? So even that's enough. I think as soon as it goes about below about 20 degrees Celsius in the soil in particular, that's when they start shutting down. So
interesting.
Baltrus thinks Jersey ants are just lazy.
Yeah, well, that too.
You're allowed to say that, Baltrus. Only a few people are. Okay. Anyway, anyone else in the Hangout have a question?
Yeah, I've got a question. Can you hear me?
Yeah.
Hi, Jesse here. First microbe seminar. Awesome, really fun. So thanks for that, first of all. But yeah, maybe just to follow up on the Clostridia and the low diversity in the natural sample versus the wild sample. So first of all, maybe just clarification. You showed there's 2 stacked bar plots. So this is like n equals 1 for each of those.
Yeah. So we've done more colonies than just the one, but obviously we want to increase that. I sort of chose to just do a one-off for the sake of visualization. But
Okay, but that is sort of seems like the general pattern.
Seems like the general pattern.
Yeah. So it is really surprising, right, that the sort of natural one is less diverse than the one from the lab.
Well, at that family level, right? I think there's a lot of diversity further down, and what we've been finding from our more the isolate work is that at the strain level, I think those Enterobacteriales are actually, they have less diversity because there's only a few mutualist lineages that are persisting through. So one of the things I'm trying to get Kevin to do is to do the oligotyping experiment, right? And at pushing his data as far as it'll go, right? At that low level, which is more diverse, I think that's going to be really interesting.
Yes, definitely. I've been I would advocate oligotyping for sure.
Yep.
As someone who's interested in sort of fine scale diversity, who is now starting to play with 16S data and being, what is this nonsense? But oligotyping is great. I would do that.
And we've got some metagenomes going on as well. So looking at mapping for SNPs and things like that as well.
Mm-hmm. Mm-hmm. Cool. I've got another question too. But I don't want to be greedy. If there's other questions in the
go for it.
Okay. All right. Well, so yeah, my other question is about, you kind of put forth those hypotheses about the I guess, for lack of a better term, I'll call it, the distribution of the secondary metabolite genes, so the kind of pan genome of secondary metabolite genes,
right.
And the distribution, and is it balancing selection? Is it neutral? Have you tried fitting any models to that? I know that Joshua White's, and there's another recent model from Jukka Korander in Finland and people like that, kind of modeling, showing that you can really account for a lot of flexible genome frequency distributions just by a neutral model. So I mean, have you thought about that, or?
Yeah, that's something that we'd really like to do. One of the problems with these data, I mean, there's two problems with the data. One is that because the genomes are so GC rich, it's difficult to get good assemblies. And so we're finally just getting to that point where we're doing it. And actually, while I was with Cameron, published a paper that showed that these gene clusters in particular are prone to blowing into pieces. And so that's one problem.
Yeah.
The second problem is that I think it's an open question of how do you say that a biosynthetic gene cluster is orthologous, right? It makes the same molecule. And one of the reasons is you can have very small tweaks in the sequence. Depending on the type of gene cluster, a single amino acid change can make an entirely different molecule with new bioactivity.
Yeah.
So I've got some computational data that's unpublished, but looking at that problem, and I think that we're waiting for the methods to advance still a little bit before we can really get to it. But it's a great idea. I'd love. That's the direction we're heading for sure.
Mm-hmm. Cool.
Well, Jonathan, as much as Maya offered Florida as a sampling location, I should say my front yard and our local state park that's full of pine trees might be a good place for you to come sample if you ever need a spot.
All right, we're there.
All right, and be careful being in New Jersey woods with spoons and needles, because, dude, that's like a natural setting for that thing.
Oh, come on, we all have stories about that.
We were more terrified in Florida because we had to go down during hunting season. So we're all there in our blaze orange and meeting all we were told we were only allowed to go in between like 9 and 3 when the gunfire was lessened. So Jersey was better than that, at least.
You guys don't have the,
stop the YouTube broadcast. If anyone wants to stay and hang out and chat more, please do. But I'm going to stop the YouTube broadcast. Thank you so much, Jonathan, for doing this, and we will be bringing you another micro seminar next month.
Thanks.